Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss a very special staining method, that is gram staining. It is a very important, widely accepted method and is carried out in almost every microbiology lab for identification, preliminary identification and observation of morphology of bacteria. What is morphology? Observation of the exterior figures or exterior organs of something. So by this method we can identify whether our bacteria is gram positive or gram negative, and we can observe the morphology of bacteria or the outer part of bacteria. So basically, what is the meaning of staining? It is in layman language, it is coloring of something. Okay, so why it is called as gram staining? Because Sir Hans Christian Gram discovered this process. So after his name we termed this process as gram staining. So it is a differential staining technique used to differentiate or categorize bacteria into gram positive and gram negative group according to their cell wall composition, because gram positive and gram negative bacteria have different cell wall composition. Okay, and it is most widely used important staining technique in bacteriology. What is bacteriology? It is the study of bacteria. Especially this method is used in medical bacteriology. Okay, in medical field it is most widely used. It is generally the first test performed on bacteria during their identification. So after isolation of bacteria and after isolating the pure culture of bacteria, we have to identify it and observe its morphology. So it is the first step carried after the isolation of pure culture of bacteria. Next, this staining technique uses two stains, two color. One is crystal violet, which is called as primary stain, and safranin, which is the counter stain. So here in this method we uses two stains. Those bacteria with gram positive cell wall will retain the primary stain, and at the end of the process it will appear as violet or purple, and these bacteria are termed as gram positive bacteria. But the bacteria with gram negative cell wall cannot retain the primary stain, and it will take the safranin or the counter stain, and finally after the end of the process it will appear as pink or reddish pink color. Okay, so this technique was first introduced in 1884 by Danish bacteriologist. Bacteriologist means the scientist who carried out the experiment on bacteria. Okay, so he was a famous bacteriologist, Hans Christian Gram. So he discovered this technique in 1884. He developed this technique to identify bacteria causing the disease pneumonia. Later it became a popular method to classify bacteria into gram positive and gram negative group or types. So what are the requirements for gram staining technique? One is sample bacterial colonies or suspension. So at first we require one bacterial colony, then a gram staining kit, and that kit have many reagents like crystal violet, safranin, acetone, then glass slide, then inoculating loop, using which we can inoculate bacterial colony on the slide, glass slide, then Bunsen burner, then staining rack, then wash bottle, and inside that we have our distilled water or generally tap water, then a microscope to observe the bacteria after staining. Then gram staining reagents. What are different kind of reagents we are using in gram staining procedure? One is primary stain called as crystal violet. It is an intensely purple colored organic compound. Chemical name of it is called as triphenyl methane dye. It is also known as hexamethyl rosalin chloride or methyl violet 10B or gentian violet. Okay, in gram staining it is used as basic dye in the ionized form of Cb plus and chlorine minus. So it will ionize to form Cb plus and Cl minus. It provides violet color to gram positive bacteria. Then we will use mordant, which is called as grams iodine. Okay, and it is an aqueous solution of iodine and potassium iodide used as mordant in gram staining. It will interact with Cb plus of crystal violet and form crystal violet iodine complex or CVI complex, which gets trapped in the dehydrated peptidoglycan layer of gram positive cell wall. So some bacteria have very thick peptidoglycan cell wall, and generally they are called as gram positive bacteria, and some have very thin peptidoglycan layer, and they have a very thick lipid layer, outer layer, which is basically made up of lipid, and they are termed as gram negative bacteria. Okay, so gram positive bacteria create a complex called as crystal violet iodine complex, which gets trapped in their thick peptidoglycan layer. Okay, next, decolorizing solution. It is either acetone or ethanol. Okay, next we will use counter stain, which is Safranin. Okay, and it is a red color counter stain. So decolorizer dissolves the lipid content which is present in the outer membrane of gram negative cell wall, bacterial cell wall, and increases its permeability. Okay, but in gram positive cell wall, the decolorizer dehydrates the peptidoglycan layer and traps the CBI complex within the cell. So the decolorizer cannot wash out the peptidoglycan layer of gram positive bacteria. It will only wash out the lipid content of gram negative bacteria. Then after washing out the lipid content of gram negative bacteria, it can retain the Safranin or the counter stain, but gram positive bacteria cannot take the Safranin or counter stain because they have a strong CBI complex formed. Okay. At first, we have to create a smear of bacterial colony on a clean glass slide, then heat fix it, and then we have to put the crystal violet solution over that fixed smear. After 30 to 60 seconds, we have to rinse that crystal violet with running water. Then we will put the iodine solution over it, and we have to leave the iodine solution for next 30 to 60 seconds. Then we will put a decolorizer, which is generally ethanol or acetone. Then we have to rinse that using distilled water. Then we will pour the counter stain, which is Safranin. Generally we use Safranin as counter stain. Then we have to leave it for next 30 to 60 seconds and finally wash with gentle running water. Then we have to dry it gently using a tissue paper or simple by blow dry method. Then after 1 minute we will observe this slide under the microscope. Okay, so it is the overall process of the gram staining or diagrammatic representation of the steps of gram staining. At first, primary stain added to the bacterial colony or bacterial smear which are present on the glass slide. then iodine, then alcohol as decolorizer, then finally the counter stain, and finally some bacteria will appear as pink, some will appear as purple or violet. Next, what is the basic principle behind it, or what is the science behind it? Gram staining and differentiation are based on the differences in the cell wall structure and composition of these two groups of bacteria. Bacteria having cell wall with thick peptidoglycan will resist the decolorization of primary stain and appear as violet or purple because they create a strong crystal violet iodine complex to which the decolorizer cannot wash out. Bacteria having a thin peptidoglycan layer with less cross linkage lose primary stain during decolorization and gain the counter stain and appear pink or red color, and that gram negative have a thick lipid layer or outer layer with which is made up of lipid, and that lipid will wash away during the decolorization process, and finally it will retain the counter stain or safranin and finally appear as red color or reddish pink color. In an aqueous solution of crystal violet dye, their molecule dissociate into Cv plus and chlorine minus Cl minus ion. These ions easily penetrate into the cell wall component of both gram positive and negative bacteria. Cv plus interact with negatively charged component of cell wall. When gram iodine added as mordant, it will interact with Cv plus to form Cvi complex within cytoplasm and cell membrane and cell wall layer. When decolorizer or decolorizing solution is added, it interacts with lipid in the cell wall, and the outer membrane of gram negative bacterial cell wall is dissolved, exposing the peptidoglycan layer. The peptidoglycan layer is thin with less cross linking within gram negative cell wall, hence it become leaky, and this causes cell to lose most of the Cvi complex. Whereas in gram positive bacteria there is no outer membrane, they have only a thick peptidoglycan layer and with higher cross linkage. So decolorizing solution dehydrates the peptidoglycan layer, trapping the Cvi complex inside the cell wall, and the bacterial retain purple color or violet color of crystal violet because this gram positive bacteria developed a very strong Cvi complex. Next, when counter stain positively charged safranin is added, it interacts with three negatively charged component of gram negative cell wall and membrane, and the bacteria become pink or red. Whereas there is no space to enter inside the dehydrated gram positive cell wall due to a strong Cvi complex and dehydration in gram positive one. Hence safranin cannot take the stain of red or pink, and gram positive bacteria reveal the purple or violet color. So it is the diagram showing the gram positive bacterial cell wall structure with a very thick peptidoglycan, and it is the gram negative bacterial cell wall structure with outer membrane which is lipopolysaccharide in nature or with a high lipid content and a very thin peptidoglycan layer. Okay, so it cannot make a strong Cvi complex, and most of the Cvi complex will wash out during the decolorization process, and it finally retain the pink color of the counter stain safranin. But here the gram positive bacterial cell create or developed a very strong Cvi complex, and they cannot retain the counter stain or safranin. So finally it will appear as purple or violet, which is the color of the crystal violet. Next, application of gram staining. It can be used in research to classify bacteria into gram positive and gram negative. It is used in diagnostic lab for identification of the pathogen, used in hospital or in medical field for choosing spectrum of antibiotic for treatment before complete identification of bacteria, used to study the morphology or exterior features of our bacterial cell. So these are various applications of gram staining. So it is all about your gram staining procedure. So I hope you will like this and will get benefit from this video in your examination. And after watching this video, if you liked it, please hit the like button and please share with your other friends, and please subscribe to my channel Sunanda's tutorial, and please stay tuned to get more important videos like this. Thank you.